Assalamualaikum warahmatullahi wabarakatuh, and a very good day for everyone. This is a pre-recorded lecture for tutorial week 9, and the title for this week tutorial is on biological molecules. Okay, part 2. Okay, it's regarding protein, lipids, and also the nucleic acids. Okay, we already covered on week 8 last week. Tutorial part regarding carbohydrate and also only carbohydrate. Okay, so in this week, okay, about lipids, okay, proteins, and also nucleic acids. Okay, the question, we have 5 total questions. Okay, the first question is distinguish between saturated and unsaturated fats. Okay, number 2, distinguish, okay, secondary and the tertiary protein structure. Number 3, differentiate between DNA and also RNA. Number 4, you have to compare 3 types of RNA. And then number 5, complete the following chart. Okay, what are the name of the macromolecules with this monomer and the function of each molecules. Okay, so for this question, for question number 1, let's see the answer. Distinguish between saturated and unsaturated fats. Okay, saturated fatty acids, okay, as we know, it consists of glycerol and 3 fatty acids, right? The difference between the saturated and unsaturated, physically we could see that saturated, it is in a form of solid at room temperature, and unsaturated, it is liquid at room temperature. Okay, the other point that we can distinguish between these two types of fatty acids is that the structure. Okay, the saturated fatty acids, okay, the fatty acids is has no kink, okay, no double bond. However, for unsaturated fatty acid, the fatty acid here, it has double bond, so it gives kink, and this causes them to be unsaturated. So again, unsaturated, they have double bond. However, saturated fatty acids, they have only single bond. Okay, so because of this, the number of hydrogen is more compared to the unsaturated. Okay, because of saturated has no double bond, so it has higher number hydrogen compared to unsaturated. Again, the presence of double bond. Okay, next, okay, the difference between saturated and unsaturated is that mostly the fats come from animal is saturated. However, unsaturated fat is fats that come from plant and also fish. Okay, and saturated increase the cholesterol in your blood. If you took this type of fats, okay, it will increase your cholesterol. However, unsaturated, it will decrease your cholesterol. Okay, and it has high melting point for saturated, and unsaturated, it has low melting point. Okay, and then you could give examples when you try to compare or distinguish between these two. For example, here it gives example of butter, okay, for saturated, and example for unsaturated, the fish oil. Okay, other than characteristic you could distinguish between these two is that it has straight chain. Okay, actually it's the same point, cuma lebih details lah kenapa dia straight chain, because it has no double bond. However, yang unsaturated, it has have a kink, okay, or band, because it has double bond. Okay, just it's the same meaning, tapi dia boleh break into 2 points. Okay, alright, that is the comparison between saturated and unsaturated fats. The best for you to explain the answer is by making a table to compare between the 2 points, the two components. Alright, next, number 2, you have to distinguish. For protein, we have primary structure, secondary, tertiary, quaternary structure, right? And usually, for your information, the structure has been described in lecture before. The structure is distinguished because of the bonds present, kan? Kalau primary structure tu hanya adalah polypeptide bond, okay, among them. Okay, secondary structure dia ada hydrogen bond, okay, that cause them to have shape of alpha helix and beta plated sheet. Right, for tertiary structure dia ada disulfide bridge, hydrocarbon chain, ionic bond. Okay, the more presence of bonds give them more complex structure. However, quaternary structure, bila you gabungan of numbers of tertiary structure, dia akan bentuk a quaternary structure. Okay, now to distinguish between secondary and tertiary structure, according to this answer, dia ada 3 points lah yang dia boleh compare. Okay, with two, one of them is example. Okay, number 1 is that the secondary structure dia ada folding of the peptide chain. Okay, membentuk alpha helix and beta plated sheet, and the bond present because of this shape is formed because of the presence of hydrogen bond. And the example of the secondary structure is keratin lah, macam rambut kita tahu it is alpha helix here. Okay, so this is gives them this structure of secondary structure. Okay, next, because of the hydrogen bond. Next, the tertiary structure, it is a 3D structure. Okay, to form globula, and the bond presents the hydrophobic interactions, hydrogen bond, disulfide bridge. Okay, and the example is enzymes. The best example for tertiary structure, enzymes. And the enzymes is as you already know, is a form of protein, right? And this enzyme is example of protein, and dia boleh denatured and renatured. However, albumin is example of tertiary structure of protein, di mana dia boleh denatured, however it cannot be renatured. Macam telur lah, this is example of telur, di mana you dah masak telur tak boleh kembali kepada yang asal lah. Okay, this is example of albumin is example of in protein that can be denatured but cannot be renatured. Okay, next, this differentiate between DNA and RNA. Okay, DNA and RNA is a part of nucleic acids. Okay, DNA, as you already know, okay, the structure of both memang kena ada. Kita tahu in nucleotide dia apa? Mesti ada sugar. Okay, and dia mesti ada a nitrogenous base. Okay, and also mesti ada phosphate group. These are the monomer for nucleic acid, which is the to call as nucleotide lah. Nucleotide. Okay, nucleotide is the monomer for DNA or RNA. Okay, and this the difference between DNA RNA, okay, di mana for the nucleotide itself, DNA dia punya presence of DNA dia punya structure, OH dia ada dia tak ada oksigen, dia hanya hydrogen ion atom. However, kalau RNA dia ada OH lah, OH, okay, dekat carbon number 2. Okay, carbon number 2 ni carbon number 1, ni carbon number 2. Kalau DNA dia punya carbon number 2 ada hydrogen atom, tapi RNA, this is RNA, dia punya carbon number 2, okay, dia ada oxygen hydroxyl group. Okay, however, for DNA, the carbon number 2 hanya ada hydrogen atom. Okay, that's why dia panggil deoxyribose, because dia kekurangan oksigen. Okay, deoxy. Tapi RNA dia ada oksigen, so kita panggil dia only ribose lah. Okay, the sugar dia ada OH, so the call is ribose. However, for DNA, the lack of oxygen, so the nucleotide we call them at the lack of oxygen atom at the ribose sugar, so kita panggil the molecule as deoxyribose nucleic acid. Okay, so the differentiate between DNA and RNA is that the pentose sugar. First point is the pentose sugar. The DNA is deoxyribose, lack of oxygen. RNA have oxygen, so kita panggil ribose. Okay, next, base composition dia. Okay, untuk bases dia lah, bases juga. Kalau DNA, bases yang terlibat ATCG, adenine, thymine, guanine and cytosine. However, for RNA, the AUCG, adenine, uracil, guanine and cytosine. Okay, other than that, the components that different, the factors that different between this DNA RNA is that the strand for DNA is long and it is double strands. Okay, however RNA it is short and single strand, because the long of strands of DNA yang double helix ni, okay, akan kekal pada pada kita punya kromosom lah. So very long strands, okay, yang mana ada they carry genetic information pada our chromosome. The function for the DNA is that carry genetic information. However, RNA for protein synthesis and found in many nucleus for DNA. However, RNA mainly found in cytoplasm. Okay, kebanyakan memang dia punya RNA banyak pada cytoplasm, di mana fungsi untuk protein synthesis. Type dia DNA lah, tak ada, tapi type dia tak ada types DNA. However, RNA they have 3 type: mRNA, rRNA and tRNA. Okay, next question number 4. Compare 3 types of RNA. So RNA tadi yang kita tahu dia made of rubber sugar. So dia ada type of bases adenine AUCG, adenine uracil cytosine guanine, and they consist of 3 types. Okay, so the 3 types ni dia ada different shape lah. Yang RNA the long strand, RNA the structure component of ribosome, it looks like ribosome, okay, macam burger, okay, it's a looks like ribosome, and tRNA dia is a transport RNA, okay, dia ada bahagian di mana base akan bergantung pada dia. So this is tRNA. Okay, so they have 3 types. The mRNA is a long single strands of RNA. RNA is a structural components of ribosome, and tRNA is the smallest of RNA. Okay, then after you discuss of its shape, focus on the function of the RNA itself. Okay, for mRNA is used to transcribe the genetic code, mana berlakunya transcription inside the nucleus. Walaupun dikatakan pada ada RNA is mostly found dekat cytoplasm, tapi actually mRNA ni terhasil, okay, dekat dalam nucleus. Okay, so actually because of the transcription process, mRNA pun terhasil dan dia berada pada nucleus. Selepas dia berada dalam nucleus, this mRNA dia akan keluar daripada nucleus pergi ke dalam cytoplasm, di mana adanya rRNA dan juga tRNA. The function of the function of rRNA is that to provide tempat untuk polypeptide assemble. Kat sinilah berlakunya proses of formation of polypeptide, membentuk gugusan protein, okay, di mana polypeptide chain akan berlaku menghasilkan protein structure, primary structure tadi. Okay, dan ni berlaku pada cytoplasm. Okay, so actually rRNA dia adalah sebagai site lah, site for polypeptide assemble. Okay, and tRNA is function is to convert the DNA information into polypeptide sequence. Okay, di mana dia akan membantu dalam pembentukan protein, di mana dia akan convert the DNA information which is from the mRNA membentuk protein. Okay, convert the information into polypeptide sequence. Okay, so it also occur dalam cytoplasm. Okay, berada pada dalam cytoplasm. Okay, next, after you discuss the function, you can compare between the RNAs, okay, according to its other description is that here mRNA, okay, carry the instruction on the sequence of amino acid from DNA nucleus to the ribosome in cytoplasm. So dekat nucleus tadi kan berlakunya transcription kan, berlaku pada proses transcription is that dia akan pindahkan maklumat daripada DNA tadi pergi ke mRNA supaya dapat dibaca di dalam cytoplasm nanti. Okay, and then the tRNA is used to pick up the amino acid in the cytoplasm dan membawa membawa amino acid. Okay, amino acid dia akan the tRNA nanti akan bawa the amino acid pergi ke rRNA untuk translate. Kan dia akan translate membentuk polypeptide. Okay, tak apa, madam akan share video untuk you tengok saja. You tak perlu pun macam in details understand the process of transcription, translation yang melibatkan these three types of RNAs. Just you tahu apa lokasi dia, macam mana bentuk dia, kat mana kita boleh jumpa. Okay, dan actually apa kepentingan these three types ni of RNAs dalam penghasilan protein. Okay, nanti kita akan watch the video. You don't have to memorize or understand very in depth the process of transcription and translation. Just tahu the shape, okay, and as mentioned here lah, and functions, okay, of the RNAs. Okay, next, complete the following chart. Okay, these are all macromolecules that been described in chapter 4. Okay, first is the carbohydrates, second lipids, third proteins, and fourth is nucleotides. Okay, so the macromolecules here, the monosaccharide, monomer, the monosaccharide. So what is the macromolecule? The macromolecule will be the carbohydrates. Okay, and then you have to know the function of the carbohydrate to provide the energy source, okay, juga such as support in plant and fiber in human diet. Okay, then dia bagi soalan macromolecule the lipid. So what are the monomer? The monomer for lipids adalah glycerol and fatty acids. Okay, then for number 3, proteins, dia tanya what are the monomer for proteins. Okay, the monomer adalah amino acids and the function is as a structure, movement, membentuk muscle kita supaya kita boleh bergerak. Dia boleh menghasilkan enzim, dia boleh hasilkan hormon, dia boleh banyakkan rambut kita juga untuk catalyze kan enzim tadilah. Okay, and hormone signaling. Okay, next, question number 4, dia bagi jawapan carry genetic information. So what are the macromolecules? Adalah DNA lah. Okay, DNA, the macromolecules dia adalah nucleic acids. Okay, and then the monomer dia nucleotides, nucleic acids, silap ni. Okay, bukan nucleotide. Dia punya macromolecule adalah nucleic acids and the monomer is nucleotide. Okay, the function of the nucleic acids, carry genetic information and important in protein synthesis. Okay, alhamdulillah we finish answer all the 5 questions of tutorial questions in from week 8. Okay, from week 9 for week 9. So let's stay focused and okay, and all the best. Thank you. Don't forget to upload your answer inside the future link. Okay, thank you. Assalamualaikum.